I want to thank the organizers for the invitation and to tell you about some of our various latest work. It was really nice to have Abigail speak right before, because I can eliminate a lot of the introduction, because what I am going to focus on is what we're learning about the role of satellite cells in muscle. And fortuitously, the same little cartoon, which originally was developed by Pete Zammit in one of his publications, which is, I really like it because it does show the classic muscle structure that everybody has had through all of their education. But for the first time, or one of the nicest illustrations, he actually included the satellite cell, as Abigail said, underneath the basement membrane, butted right up against the sarcolemma of the muscle fiber in this satellite position, which is why Morrow called it a satellite cell. And that the study of these cells has really been augmented tremendously in the last 5 years, almost 10 years now, with the advent of the antibody against PAX7. So as Abigail pointed out, satellite cells express high levels. PAX7 is a transcription factor, and it's absolutely satellite cell specific in muscle. And it's a wonderful antibody for any number of purposes. And it really does identify satellite cells very convincingly, either on isolated single fibers or in cross-section. And I thought I would just show you, this is just a picture from a mouse muscle, although human muscles look much the same. And you can see that it is really very easy to count satellite cells and express them per muscle fiber with this antibody, which is very specific and allows one to start to look at the behavior and the location and the number of satellite cells in response to a number of conditions. And what we know, based on a lot of years of evidence, but then most recently definitively using a model where we can actually kill satellite cells, a number of groups have shown that, in fact, definitively satellite cells are the muscle stem cell. And that when you damage a muscle, and now most of this work, well, all of this work is done in rodents, because you inject cardiotoxin or something nasty into the muscle, the whole muscle degenerates, which you obviously can't do in humans. But when you do that, you see that satellite cells become activated, they migrate, they'll align, they'll fuse, and they'll reform a muscle fiber. And that they are the stem cell responsible for doing this. Now, this has been very clear, and so we wanted, though, as opposed to studying muscle regeneration, we have been interested in using more physiological models, some that mimic exercise in the rodents better, in mice. And so we've done various models, such as models of aerobic training, and luckily in mice, especially in the background that I'll be telling you about, black 6 mice love to run. And if you just give them access to a running wheel, they'll run 15 kilometers a night, every night, if you let them. And so one can study the role of satellite cells in aerobic training, and I don't know if you can equate that to elite training, but they run all night long if you let them. And what we've also been studying are trying to develop models, not develop, they've been around for a long time, but we use models of resistance training. And since it's harder to get mice to resistance train, we use a surgical model there, synergist ablation, where we cut the Achilles tendon and remove a portion of the gastrocnemius muscle and the soleus muscle, which promotes a functional overload of the plantaris. And so it hypertrophies, and that's the resistance training model that we used. And the mouse, the data that I'm going to tell you about today is using a mouse which Abigail again referred to, and it's one in which we specifically deplete the satellite cells in adult muscle. And here is the model, and that is that you use this PAX7 promoter, which again is just expressed in satellite cells, and you drive a Cre recombinase. So this is a viral protein, which is a nuclease, and so it's modified such that it has it's a transgene forming a fusion protein with a modified estrogen receptor, which doesn't bind estrogen, but it does bind tamoxifen. And because this modified estrogen receptor blocks the Cre recombinase activity, it's not active unless you treat the mice with tamoxifen. Okay, so that's silent under normal conditions. We crossed that mouse with a mouse in which a very strong constitutive promoter was driving the diphtheria toxin A gene, which is very toxic to cells. However, between the promoter and the gene, we inserted a stop codon which has the LOX-P sites, which is recognized by the Cre recombinase. So you just cross these mice, and we generate the PAX7 mice. And then when the mice are full-grown adult mice, 4 months old, these are, the long bone growth has stopped, so they're full-grown adult mice. We treat them with tamoxifen for 5 days, an injection every day, intraperitoneally, and then we do a 2-week washout. And the data I'm going to show you then is doing the synergist ablation surgery at that point and then following them. Now, this tamoxifen treatment very effectively depletes satellite cells, and surprisingly to us is that the satellite cells remain depleted throughout the life of the mouse, which allows us to do a lot of experiments in terms of the role of satellite cells in normal muscle maintenance and muscle aging, because they don't recover. They remain greater than 95% depleted. And we test that in all of our animals when we're doing various experiments by injecting cardiotoxin, or I'm showing you here barium chloride. We've moved to get away from the toxins. And what you can see is this is a week after injection of cardiotoxin, and you see muscles regenerating as indicated by the nice little centrally located fibers. However, in the tamoxifen-treated animals, regeneration is completely impaired, so that it's clear the satellite cells have been depleted. However, we find, and so in our hypertrophy model, that is the synergist ablation model, what we see here is that satellite cell function is also impaired. Because as you can see here in a cross-section, one can quantify myonuclear accretion. So with hypertrophy, normally satellite cells do fuse in and add a nucleus to a growing muscle fiber. That's clear in mice and in humans. And one can see that in the vehicle-treated animals. However, in those that have been tamoxifen treated, one can count the myonuclei. Here, a section is stained with an antibody against dystrophin, which shows the sarcolemma of the muscle fiber, and then just DAPI, which is a dye that intercalates into DNA, so it stains all nuclei. So one can count nuclei inside the fiber compared to ones, I'm just showing you examples outside the fiber. And if you count that, you can see there is, in fact, myonuclear accretion normally with hypertrophy. However, in the animals that have been treated with tamoxifen and SA, that means synergist ablation 2 weeks, though they've been overloaded for 2 weeks, you see that myonuclear accretion is absent, so that these animals are able to grow without adding myonuclei. And that is, though, up to a point. And what I want to tell you about today, the 2 different phases, or what we think are 2 different phases of hypertrophy. So early during hypertrophy with tamoxifen treatment, you see relatively normal growth. However, in the later stages, at 4 weeks and 8 weeks, which is the latest that we've looked, you see that the growth plateaus. So clearly there's a mechanism involved in mice, and mice are incredibly adaptable little critters, and many of the things that they're able to do and adaptations that they exhibit may not necessarily be relevant to humans. But in this case, we want to try to understand what is the compensatory mechanism here that allows the muscles to grow without adding a myonucleus. And then I'll tell you some about what we think is going on here in the later stages of hypertrophy. So some very preliminary results that what we think might be going on here is that, and first let me tell you how we do this experiment. So again, just illustrating the experiment, we do in the 4-month-old mice, we tamoxifen treat, we let it wash out so that all of the tamoxifen is gone, we do the synergist ablation surgery, and then 2 weeks later, we sacrifice the mice. In these sets of experiments, what we do 5 hours before sacrificing the mice, we've injected IP with a uridine analog, ethylene uridine, which using click chemistry, you can actually quantify it in cross sections. And we did a bunch of time course experiments and saw that at 5 hours is the best time to be able to detect the nascent labeled RNA within the nucleus. So it hasn't all yet diffused out into the cytoplasm. But if at that time, if you use click chemistry it's a biotin-avidin based system, one can now visualize the nuclei that are most highly expressing the uridine or accumulating the uridine analog. And in these pictures, they're pink. The nuclei are pink, which are labeled. And this probably mostly represents ribosomal RNA, so maybe a reflection of ribosomal biogenesis. And what we found was that at this 2-week time point, the percentage of nuclei that are labeled and the intensity of the labeling of the nuclei, which I hope you can see here inside the fibers, is much higher in the tamoxifen-treated animals. So that what we think is happening, somehow the nuclei can communicate with each other, and that when there are no satellite cells to fuse in, the existing myonuclei can ramp up their transcriptional activity, and that can promote as much ribosomal biogenesis so that they're not at their max. However, when there isn't a nucleus, aren't satellite cells to fuse in, or that when there aren't satellite cells to fuse in, they can ramp up their transcriptional capacity, so that normally, although they are increasing their ribosomal biogenesis followed by increase in protein synthesis, they're still, this is not at the max, is what I would say. And so we're trying to look at this mechanistically and try to understand how the fibers may be compensating and able to increase their biosynthetic capacity in the absence of addition of new nuclei. So that's the kind of experiments that we're trying to pursue, trying to understand this compensatory mechanism. But now I want to focus on these later, the sustained, the ability to sustain growth in the absence of satellite cells. And as I said, it plateaus, and you can see this here by fiber cross-sectional area, that you see an increase in average cross-sectional area, which is blunted at the 8-week time point. And we started down a line of experiments thinking, well, this is somehow reflecting this a limit of the myonuclear domain, and that the fiber's growing, and it gets to a point without addition of another nucleus, perhaps the fiber can't grow anymore. We're still trying to explore that, and we have some other mouse models that we've developed and gotten from other people, where we can. One of them is an animal in which a gene has we can specifically deplete a gene using the tamoxifen approach, which inhibits the satellite cells from fusing, so they're still there, but they can't fuse. And so we're trying to understand what might be limiting the growth in this case. But by looking at the muscles out at this 8-week time point, we think we have a new hypothesis on what might be going on when you deplete the satellite cells. And that's shown here. Again, we think that the satellite cells, in the absence of satellite cells, the muscles become fibrotic. So that here, just showing two different ways to visualize the extracellular matrix around the fibers, that once you deplete satellite cells and then you impose this overload on the plantaris muscle, that you can see that even just the absence of satellite cells without overload in a sham animal, you see a significant increase in fibrosis, which is really exacerbated with overload. So that what we think is happening is that the satellite cells are doing more in the muscle than just fusing in a new nucleus, which was what they're doing when the fiber grows, but they're actually involved in controlling the extracellular environment. And that makes sense to me, because the increase you see in the number of satellite cells with overload is way more than the number of nuclei you add in. So that because there's this huge increase with overload, if they were all to fuse in, they would, you just end up with a big bag of nuclei, of fiber, so that there's a bigger increase in what you need to add nuclei to a growing fiber. And we think that they're involved in signaling. So to try to study this, as we designed and we did some in vitro experiments, because from the mouse, one can easily isolate satellite cells and one can isolate fibroblasts from the same muscle. So we did that, we put them in vitro, and then did some co-culture experiments. So that's shown in the next slide. I should say just that this fibrosis that we see, we think reduces force generation in the muscle. If you look at single fiber function, it's not different between the animals. So the individual fibers, even though they have fewer nuclei, generate as much force, but the specific force of the muscle overall is impaired. We think probably because there's less contractile tissue there and there's more connective tissue, more fibrosis. So getting back to the in vitro analysis, this is how it's done, where, as I said, we isolate the satellite cells, and once, sort of convention in the field, once they're isolated out of their satellite position and they begin to divide, they're now not really stem cells or satellite cells anymore, they're myogenic progenitor cells, so MPCs. One can isolate those, as I said, from the muscle, and we put those on a little insert, and this is a membrane so that media is freely diffusing between, and co-culture those with the fibroblast from the same muscle, or as a control, just co-culture fibroblast with fibroblasts to account for the depletion of nutrients in the media. And then, subsequently, we isolate these fibroblasts after 24 hours of co-culturing and just do real-time PCR to look at extracellular matrix gene expression. And this is what you see. And that is that fibroblasts that have been co-cultured with MPCs seem to have a repression of collagen gene expression, but a number of components of the extracellular matrix. So we think that the satellite cells and the activated satellite cells, or MPCs, in vivo are signaling to fibroblasts and regulating their extracellular matrix gene expression. Now, of course, we've been very interested in how is this mediated. It appears because of this co-culture that it's a secreted factor. So we've done experiments with conditioned medium then. Just take the MPCs, allow them to condition the medium, and put that on fibroblasts. And what we've also done is to, with all the interest now in exosomes, we've actually conditioned media and isolated exosomes from MPC conditioned medium and put that on fibroblasts. And what we see is the following, is that both conditioned medium and MPC-derived exosomes both seem to inhibit collagen gene expression in fibroblasts. So we think this is a real mechanism that's ongoing in muscle. And what we're working on, once you start to talk about repression of gene expression, of course, microRNAs come to mind. MicroRNAs have been shown to be packaged in exosomes. And of course, they bind to messenger RNAs and either destabilize them and or inhibit their translation. And so this is a very preliminary experiment. What we've done is isolated the MPC exosomes and labeled them with acridine orange, which is a dye which just intercalates into RNA. We've washed those exosomes extensively and then just incubated them for 2 to 4 hours on fibroblasts, and then look at the fibroblasts. And what you see is there's now acridine orange in the fibroblasts. So we think this is a microRNA-mediated event. And we're following up on that and trying to identify, we're profiling the microRNAs that are contained within exosomes and trying to identify what then the targets might be. But this is all a very new line of research for us. And so I can't tell you what targets. And some of the microRNAs we found in the exosomes may directly target collagens. Some may target regulators. So we think this is a whole new avenue of research for us. But so what I want to do here is just real quickly conclude this part, just our work on the mouse, and that is that we think that the existing myonuclei have the capacity to upregulate transcription, and that this is sensitive to the DNA content of the muscle fiber, that hypertrophy is limited in response to not long-term overload, and that is associated with fibrosis, and we think that this may even actually be what's limiting the growth long-term. And we're doing experiments to try to test that hypothesis. And that, as I said at the end, that we think that satellite cells or activated satellite cells, MPCs, actually regulate the muscle fiber environment and may facilitate remodeling by regulating extracellular matrix gene expression in fibroblasts. So what I want to do real quickly, just for the last few minutes, is tell you about some work that we've done in humans, some of our human exercise studies, which were designed initially for a completely different reason, but now they're starting to inform our mouse studies and causing us to think a little bit differently and having us design some different kinds of experiments in the mouse because one can do more mechanistic studies. So we had started and did some years ago just an exercise, cycling exercise study in middle-aged sedentary individuals with the idea of studying effects of exercise and obesity on insulin resistance. So that was the point of the study. But when we actually started analyzing the data, just as an aside, a graduate student looked, and we saw that the individuals showed a modest amount of hypertrophy with this cycling. They worked very hard, these individuals. This was a 3-month training study, 3 days a week. For an hour, they came in on a stationary ergometer and at approximately 70% of heart rate max for the last several months. And so you can see that modest but significant increase in fiber cross-sectional area, and here by just bending the fiber size, you see the characteristic switch to the right, so that you're seeing an increase in muscle fiber size. And we looked at that by fiber type. Just using the standard battery of isoform-specific myosin heavy chain antibodies, those that recognize type 1 myosin 2A, and also 2X. And in our hands, at least in these individuals, we never see any, in these middle-aged sedentary individuals, any pure 2X fibers. They're all 2A, 2X hybrids. So we did fiber cross-sectional area by fiber type. did that analysis, and here's what we found, is that both the type 1 and the type 2A fibers increased in size. Although there was a slight trend, there was no significant change in the cross-sectional area of the hybrid fibers. So what we wanted to do next was to look at this hypertrophic response and the role of satellite cells. And one can do that, again, immunohistochemically on cross-sections from biopsies. I should say these are all biopsies from the vastus lateralis, as you probably guessed. Now here, doing different antibodies on the same section, here is the myosin, type 1 myosin antibody. So the black fibers are the type 2. We didn't add the type 2 antibody here for reasons to do with the second antibody so that we could do this all in the same section. This is an antibody against laminin, so the basement membrane, which, as we mentioned, the satellite cells reside within the basement membrane, and here's the PAC7 staining, and here's just DAPI, so you see all the nuclei. And of course, this is all in the same section, so you can overlay these, and it allows you to quantify fiber type specific satellite cells. And that's shown here, just giving you an arrow. Here are 2 satellite cells, so there were only 3 labeled satellite cells, right, with the PAC7 antibody from the previous slide. These 2 are obviously both type 1 satellite cells, and this is one that's associated with the type 2 fiber. So when we did this analysis and counted the satellite cells, what you see is overall there's an increase. So this this cycling training in these individuals caused an overall increase in the number of PAC7 positive cells or satellite cells. But what you can see is that was only in the type 1 fibers. So there's a preferential increase in satellite cells associated with type 1 muscle fibers in the human. That's associated with an increase in myonuclei only in the type 1 fibers. So here you can see now, because we DAPI stained, which stains all nuclei in the fiber, in the whole muscle, one can count type 1 nuclei total. Here you can see, and you can count, here's just an example of a type 2 fiber nucleus. And so one can quantify that as well. And what you see when you do that is that the increase in myonuclei per fiber is preferential in the type 1 fibers. So why is this relevant? And trying to tie this all together, the plantaris muscle in the mouse is almost entirely fast. In the entire muscle, there are perhaps 3 type 1 fibers in the entire muscle. So it's an entirely fast muscle that we've overloaded and done our mouse studies on. And this would imply that although there's growth in the type 2 fibers, in the human, now I'm going back and forth between human and mouse, that in fact the satellite cell increase and the fusion with this very modest amount of growth is preferential in type 1 fibers. So this made us rethink about our analyses, but what it would imply is that type 1 expressing muscle fibers have a much more strict requirement for satellite cell fusion and for myonuclear accretion, whereas type 2 fibers appear to be more flexible in their myonuclear domain. And this makes sense, and there's certainly lots of literature out there that would support this kind of conclusion, given that the type 1 fibers are more oxidative, they have a much higher mitochondrial content, and it could be that they really are at their biosynthetic capacity, trying to maintain the volume of mitochondria in those fibers, and that in order for them to grow, they require addition of a myonucleus. On the other hand, perhaps type 2 fibers are more flexible, that they can grow to a certain extent without the addition of a myonucleus. Now, what the myonuclear domain limit, there's a lot of work out there suggesting what that might be in humans at least, and it may be that they will reach that limit, but that there is more flexibility there. So we're going back and reevaluating our mouse work and trying to see if we can get at this question by looking by fiber type and to look at different muscles and overload them in different ways so that we can get at this, because there's certainly some indication that protein synthesis levels are higher in type 1 fibers, and there's a number of studies from a number of labs for many years which would support this sort of idea, but that we do think that different potentially cellular and molecular mechanisms may regulate growth, may regulate hypertrophy in fast compared to slow oxidative muscle fibers. So I'm going to conclude here with just an acknowledgement of the people in the lab who've done most of the work. So it's led largely by a wonderful postdoc and graduate student in the lab, Chris Fry and Tyler Kirby, but a lot of people contributed. And I always want to give a shout out to our undergrads who spend a lot of time counting and measuring and in front of the microscope, and they are just fantastic. And it's reassuring sometimes because most of the time they're blinded to the experiment, so I don't worry about that sort of thing. And this is a long-term collaboration with two of my colleagues at Kentucky, John McCarthy and Esther DuPont-Verstegen, and Chris Mendiaz helps us with the muscle functional analyses, especially the single fiber analyses. And I should say the work is supported by the NIH, both the National Institute on Aging and Arthritis and Musculoskeletal. So thank you.